Hello friends, myself Dr. Kumar, Assistant Professor, Galgotia University. Today we will discuss the topic nucleosomes and DNA packagings. The objective of this topic is to understand the architectures of the nucleosomes and the packaging of the genetic material in eukaryotic systems. The cells contain a nucleus which is surrounded by the nuclear membrane in all the eukaryotic systems you all know. And the nuclear regions in the prokaryotic cells is totally different from the eukaryotic cells. In a non-dividing cells, the nucleus is filled with thread-like material which is generally known as the chromatin. This chromatin material is made up of the DNA and the protein. This DNA is attached with the protein known as the histone protein, and some part may be the non-histone proteins and may be the acidic in nature. The chromosomes themselves are the macromolecules entities that must be synthesized, packed and protected and properly distributed to the daughter cells by the cell divisions. The large amounts of the DNA remain packed inside the cells. The packaging of the tremendous amounts of the genetic informations into the small space within a cell has been called the ultimate storage problem. The chromosomal DNA exists in the form of very long molecules which must be tightly packed to fit into the small confines of the cells. The structure of the DNA can be considered at the three hierarchical labels. That is, the primary structure of the DNA is its nucleotide sequences. The secondary structure is a double-stranded helical, and the tertiary structure refers to a higher order which folding and allow the DNA to be packed into the confined space. Now the nucleosomes. You can see that nucleosome, it is a fundamental unit of the DNA packaging inside the eukaryotes. This nucleosome consists a segment of the DNA, and this DNA remain bound in the sequence around the 8 histones proteins, and these proteins remain in the sequence, which is clearly seen in the diagrams. And that this structure is often compared to thread which is wrapped around a spools. In this diagram you can see this is the side view, in which you can see this is H1 histone protein, this is the DNA strands which surround the proteins, and this is the octamer of the histone protein, means these are the components of the histone proteins. And this is the top view, in which you can see a complete molecules of the nucleosomes. The nucleosomes form the fundamental repeating units of the eukaryotic chromatin, which is used to pack the large eukaryotic genomes inside the nucleus. The nucleosomes are folded through a series of the successive higher order structures which eventually form a structure known as the chromosomes. This both compact DNA and creates an added layer of the regulatory control which ensures the correct gene expressions. The nucleosomes are thought to carry the epigenetically inherited informations in the form of the covalent modifications of their core histones proteins. The nucleosomes' positions in the genomes are not random, and it is important to know where each nucleosome is located, because it determines the accessibility of the DNA to regulatory proteins. Now we will discuss some history and the discovery and some discoverer of the nucleosomes. So first of all, the nucleosomes were first observed as the very small particles under the electron microscopy, and the scientist who first of all observed under the electron microscope was the Don and the Ada Orleans, who observed in 1974. Their existence and the structure first of all were proposed by the scientist Roger Kornberg. The role of the nucleosomes as a general gene repressor was first of all demonstrated by the Large et al. In vitro and by the Han and the Grunstein in vivo in 1987 and 1988 respectively. The nucleosome's core particle consists approximately 146 base pairs of the DNA, which is wrapped around the 1.67 left-handed superhelical turns around the histones proteins, and also consists the two copies each of the core histone proteins, which are the H2A, H2B, H3 and the H4. Here this is a diagram in which you can see a linker DNA, and this is the histone tails, and these are the different components of the histone, which are the H3, H4, H2A and the H2B. The linker histones such as the H1 and its isoforms are the involved in the chromatin compactions. The non-condensed nucleosomes without the linker histones resemble a beads-like structure, and these beads are present on strings of the DNA when we observe under the electron microscope. The nucleosomes consist of a core particles of the A8 histone proteins, and the DNA wraps around the core, which is clearly seen in this diagram. This distance is around the 30 nanometer, and the diameter of the one histones is a 10 nanometer, and this is the strands of the DNA which wraps around the histone proteins, and the whole structures is known as the nucleosomes. The chromatosomes, which are the nucleosomes which are bound to an histone protein and separated by the linker DNA. This whole structure is known as the chromatosomes. The nucleosomes fold and form around the 30 nanometer chromatin, that is the 30 nanometer chromatin fiber, which appears as a series of the loops that pack and create around a 250 nanometer bite fiber. Helical coiling of the 250 nanometer fiber produce the chromatin structure. In this diagram, you can see this is the top view of the histone octamer, and this is a side view of the histone octamer. In this case, you can see the nucleosomes core structures in which the octamer formation has been shown, and this is the core structures in which the DNA strands wraps around the histone proteins. This table shows the compositions and different variations of the different types of the histones proteins, and in these structures you can see this is the structure of the one nucleosomes in which the octamer unit of the histones are the present, in which DNA wraps around the histone proteins and forms the nucleosomes structures. Now the histone proteins. Histone generally present as octamer form and associated the DNA and form the nucleosomes structure, and the nucleosomes remain combined with the H1 proteins and form the structure which is known as the chromatosomes. The additions of the H1 to a nucleosomes results in the protections of an additional 20-22 base pair of the linker DNA which is adjacent to the nucleosomes, and thus H1 is often referred to as the linker histones. Only one H1 subunit is present per chromosomes, unlike the core histones which are the present in two copies of each nucleosome. The DNA binding in H1 is intrinsic to the central globular regions, which contains two DNA binding sites. Now in these structures, you can see the octamer structures of the histones and the different separator, generally known as the linker DNA, which bind these octamers to each other. The diameter of this structure is around 11 nanometer. So the H1 binds only one of the linker DNA strands and the second DNA site in the histone. The H1 binds to the central region of the DNA and make the supercoiling in the nucleosomes. The histones are rich in the basic amino acids. Generally, the basic amino acids such as the arginine and lysine are present in the histones, which together make up around the 25% part of the total amino acid residues in any given histones proteins. The histones proteins are highly conserved among the eukaryotic cells. And the histones H3 and the H4 are nearly identical in all the eukaryotic cells and suggesting the strict conservations of their functions. The histones H1, H2A, H2B show the less sequence similarity, but on the whole, they are more conserved than the other types of the histone proteins. Some salt bridges between the positive charge histones and the negative charge DNA also play major role and provide the stability in DNA histone complex molecules. Now the H1, H2A, H2B are rich in lysine amino acid, whereas the H3 and the H4 are arginine rich molecules. H1 is highly unconserved and mutable proteins, whereas the H3 and H4, these are the very highly conserved molecules. According to the molecular weight, there is a particular series and the sequence in which the H1, H3, H2A, H2B and the H4 as per the molecular weight relations. And the histones proteins generally lack the tryptophan amino acid. Now some non-histones proteins are also present in the nucleosomes. In chromatin, those proteins which remain after the histones have been identified and classified as the non-histone proteins. The scaffold proteins, DNA polymerase, some heterochromatin proteins, 1 and the polycomb are commonly called as the non-histone proteins. The non-histone proteins are of the high molecular weight and approximately 1 to 1.5 lakh Daltons in molecular weight, and they are the acidic in nature and they functions as the enzymes and promotes the gene expressions.